So you want to understand why do we sleep? If yes, you are in the right place. I am starting a series where I am reading one research paper in like one big huge field. This time it's neurobiology and sleep homeostasis. So this is the first paper that I've started with because I've been obsessed with sleep when I started reading this book. So this is the culprit why I am in this rabbit hole. So yeah, so this is a very landmark paper. It has been published in Nature, and it actually decodes a very big mystery, the mystery of why do we sleep. Yes, it is a decade long, actually more than that. It is a mystery which hasn't been decoded in the cellular level. There have been a lot of studies in the like organ level of melatonin's role, different electric phases in the sleep cycle, like mentioned in this book, REM and REM sleep, sleep hygiene, and as to how you can die if you don't sleep. So yeah, all type of researches have been done, but not the cellular or the molecular research. So this is the paper which solves the questions we have. Okay, so yeah, let's dive into it. So, as I've talked in my previous video as to how to approach a paper while reading it, right? I am going to change a little things here again. This is my first time making these kind of videos, so I am, experimenting with a few styles as to how I can do it because I don't have an audience like I have during my course. I miss you all my batchmates. Okay, so yeah, uh, let's start with the broader questions and the broader concepts that are in the paper and that gonna come again and again. And if you don't understand these questions, you won't understand the figures, the results, or just anything. So I've divided it into four questions. That becomes easier because whenever a question mark comes, something happens in the brain. Okay, so what are the questions in the paper? First is the physical trigger for sleep. So, sleep again have been studied, but there wasn't any study or any explanation on what could be the physical trigger. What is some tangible method, tangible substance that is, creating the cascade event that leads to sleep, or like what organelle or like what kind of molecule reacts with what kind of receptor. Just no explanation at all. So this is the question, what is the physical trigger for sleep? Then comes what is sleep pressure. So this is a term that has been recently used, and I love this. Like I love these two words together, because it is a subjective feeling of you know um, the pressure to sleep, and it cannot be measured like the other things that are related to sleep pressure can be measured, and in at last concluded that yes, there is a sleep pressure that is causing the organism to sleep, but sleep pressure in general you cannot measure it. So um, involving the usage of this word is very it's very interesting. Okay, so the next comes that the general question of why do we sleep, and well, it's not just the book's title, but it also tells you about the evolutionary reasoning of why sleep came into the picture during the evolution and how it became an integral part of the metabolism. So yeah, and a lot of things that I'll talk about in the conclusion. Then comes the cellular mechanism. We are going to deep dive into the proteins and genes of mitochondria. So it's going to be an interesting one. Okay, so for the study, you need a model organism. So what is a model organism for this study? Like all my biology research fellows, I know you already know. It's a neurobiology study. It needs to be Drosophila. It's the superstar for all these kind of studies. So this cute little fruit fly is the subject of a study. The researchers actually divided them into two cases, which is the rested and the sleep deprived flies. So they simulate conditions to, you know, make these phases, like, I don't know, make the flies experience these states during the duration of the study. And it's an interesting experimental plan that I'll talk later. So there needs to be like an observation that the researchers must have, experience, because whenever a study is done, you need to have like a, the flashbulb moment to go into that direction. Otherwise, it won't yield any of the result for the hypothesis that you might create. So it needs to have like a little bulb on the road that shows you the path. So for this, the researchers actually characterized the transcriptomes of single cells which were isolated from the rested and sleep deprived flies, and it was observed that there was an upregulation of these transcripts after sleep deprivation. So, in sleep control neurons, which actually project to an organ, again, this is a prior study, and these researchers built on it. So, it said, like, it showed that DFBNs, which is the dorsal fan-shaped bodies, are responsible for sleep homeostasis, regulating sleep in Drosophila. And it's like a simple thing, one organ, and it's like taking care of the process of sleep. But in mammals, it's very complex. Not a single part of the brain is responsible for sleep. It's like it's a very long list, but it's mostly hypothalamus, thalamus, and the brain stem, and a lot of different centers, like almost 10 more centers that work together to build up the sleep pressure. So there is a little bit of analogy between the two, like two organisms, the mammals and the Drosophila, and this is a big win. So yeah, so this is why they tried to study the sleep pressure in Drosophila. So yeah, the superstar organ, the dorsal fan-shaped bodies look like this. It is named fan shaped because it looks like one of those Chinese fans, and it is in the dorsal part of the fly, so dorsal fan shaped body. So actually, these bodies encode proteins for mitochondrial respiration and ATP synthesis. So this was the place where everything clicked. Like, okay, so this is the center, also responsible for sleep, and also the mitochondrial respiration and ATP synthesis proteins are being coded in the center. So they see that the upregulation after the sleep deprivation is related to DFNBs, and that's where they created a very sophisticated experimental plan where they try to see a lot of different processes that happens in mitochondria during the respiration and the ATP synthesis period, and also some of the process that are the result of cell sensing stress inside inside it. Also the outside environment, but mostly inside the cells where organelle and the other signaling gives gives out the output that the cell is in a lot of stress. Okay, so the first comes mitochondrial fragmentation. As the name suggests, it is just not the fragmentation, but it is a part of the cycle where the mitochondria actually undergoes fusion and fission. So in fusion, there is an increase, a net fusion activity, and that is when the mitochondria becomes more elongated. Well, the diagram says just elongated, but it sometimes become more spherical. Also, it increases in the width, and sometimes it increases in branching as well. So a lot of times these diagrams don't show the branching of mitochondria, but they are beautiful when you see it in different microscopic images, especially when they are tagged, and yeah, it's beautiful. Okay, then comes the fission. So in this process, the mitochondria actually wants to isolate the part where mitochondria is damaged, or it experienced stress, and that is when it like kind of diverted all its resources away from certain area or certain part of the mitochondria. And when the fission takes place, there is a healthy mitochondria, and all the damaged part goes into the into a damaged mitochondrial assembly. So in this, the fission activity is increased, and you can see smaller mitochondria with lesser mass, lesser vigor in the outer membrane and the inner membrane. So yeah, and this is the part where it's easy to understand the relation between mitochondrial health and the sleep pressure. So look out for this. Okay, then comes the enhanced mitophagy. So as I've said that there is increased fusion fission activities in the sleep pressure regulation. So what happens when there is a sleep deprivation state, the mitochondria undergoes fission. Okay, so it is getting degraded and getting converted into the scratch molecules, but these scratch molecules are actually used in the biogenesis of healthy mitochondrions. So this is a cycle where it leads to more healthy mitochondria in the cell system. Okay, then comes that there is an increase in the number of contacts between mitochondria and endoplasmic reticulum. So it is quite well studied. Like, of course, not very documented part of the science, but yes, there have been a lot of great advancement in this field too, that the ER and mitochondrion are quite connected physically through channels and receptors, and they exchange a lot of material like lipids and a lot of proteins which get mm, you know, modified in ER and then sent back to mitochondria, or mitochondria wants to send out an information like in terms of a molecule, in terms of a signal molecule, and it needs to have certain changes in it to send it out of the out of the ER network into the cellular cytoplasm. So it's not a one definite path, but yeah, it has a lot of connections, and the proteins which are encoded for these exchange networks are actually SPLICs. So there is a result diagram that is going to come which will explain it better. Then comes that it creates conduits for replenishment of peroxidized lipids. So I was like really confused as to how peroxidized lipids is related to sleep, right? So everything gets connected here. As I have said that mitochondria stresses the signal, the mitochondria senses the stress, and it leads to the fragmentation or the mitophagy, right? So what happens here is that in the stress conditions outside of the cell, it could be anything like the starvation, hypoxia, or reactive oxygen species. So due to these stresses or some kind of a chemical stresses, there is a structural addition to the lipids which changes the overall integrity of the cellular membrane, and that's when there is an more exchange of substances between the outer environment and the cell. So with good things comes the bad things too, and there could be antioxidants coming which could be better for cell sometimes, but again then there could be irritants and different waste molecules that enter the cellular membrane that were first kicked out of the cell, and it creates a lot of stress conditions. That is when a misfolding of proteins happen, a lot of organelles get stressed, and it might lead to autophagy or apoptosis. So it then creates an overall distress signal for the cell, and that is how the stress inside the cell gets built up. So yeah, this is the connection for the peroxidation of lipids, and it will be explained more, it will be easier to understand when it comes in context in the later part of the paper. Okay, so here we need to keep note of 2 points, that hyperfused mitochondria actually increases the neuronal excitability and sleep, whereas the fragmented mitochondria decrease the neuronal excitability and sleep. Okay, this is very counterintuitive to what I said in my previous video, where you need not to see the conclusion of what the study has done, but this is a part where it would be easier for you to understand the understand the paper more efficiently. So that is why I'm throwing these two statements out here. Okay, so this is how they, um, you know, ran the study. They first did the single cell suspension, then it underwent flow cytometry and also single cell RNA sequencing. They also tagged a lot of these cells under un under differential control of GAL4 in this case, and well, they wanted to see the region which were encoding proteins for these neurotransmitters, which is the glutamate, which is also known as GABA, and the vesicular acetylcholine transporter. So, in the study, they saw that there was only an upregulation of the GABA, and they could not see any statistically, significant change in the acetylcholine transporter. So, what happens actually in the CNS of Drosophila is that these GABA glutamate act on the ligand gated chloride channels, and this is known for its inhibitory effect on the DFNB's postsynaptic neurons. So it inhibits DFNBs, which is a sleep regulating center, right? And this only happens when the flies are in a more awake state, where it needs to inhibit the DFNBs. Okay, so this is the first study where they saw the upregulation and the downregulation of different proteins during the sleep deprivation phase. Here you can see that the mitochondrial respiration and ATP synthesis, like the mitochondrial electron transport, which is cytochrome C to the oxygen, the whole mitochondrial respiratory chain, the glycolysis pathway, the mitochondrial ATP synthesis, coupled electron transport, and the pyruvate metabolism, everything that is related to the respiratory chain and ATP synthesis gets upregulated. So, like, the hypothesis is making sense, guys. Okay, so the remainder of the paper actually discusses the causes and the consequences of these differential expression of genes of mitochondrial proteins in the DFNBs. So this is the point where they hypothesize that the sleep and aerobic metabolism are actually fundamentally connected. And to answer these questions, the experimental logic they want to put out was that there is a beta subunit, the potassium channel in the DFNBs, which actually regulate the electrical activity of the DFNBs. And this subunit actually has a NADPH connected to it, and the electron state of this, decides or tell us about the fate of the electron in the respiratory chain. So what happens is, when there is a more demand of ATP synthesis, there is an electron leak from coenzyme Q to cytochrome C, which is a complex 4. And when there is an electron leak, these electrons, enzymatically reach oxygen enzymatically binds to oxygen, and it creates superoxide and other reactive oxygen species, which, as I've already mentioned, leads to a particular cascade effect. And during the waking time, which is the prolonged, the prolonged um, resting is is the time where the ATP demand increases, thus the leaks, and more ROS is getting produced, and the opposite takes place while the flies are sleeping. Okay, so you cannot actually measure how much electrons or how much energy is getting converted in these electron transport chains. It's very complex. So you need to introduce and like target a particular um, part of the electron chain to understand the dynamics, the energy dynamics of the whole chain, right? So what these researchers did was that they introduced heat stimulus to mimic the waking phase of um, to mimic the waking phase basically. So not just the heat stimulus, but they also use light packets to transfer the energy to these electron transport systems. So here, as you can observe, that the heat stimulus is creating the same kind of conditions when there was the baking phase. So it leads to some of the observations that shows that there will be a risk to polyunsaturated membranes, right, as I've already mentioned, and how DFNBs actually estimate the size of the mitochondrial electron leak is through counting the reductions of lipid peroxidized carbonyls at the hyperkinetic. Hyperkinetics active site is the is the potassium channel shaker. Okay, so this is one of the results where they saw the DRP1, which is the fusion protein of the mitochondria. Then comes the SPLICs, which I've already mentioned, are the connections between ER and mitochondria. And then comes the mitoQC. Mito-QC is actually how much it is undergoing like mitophagy and the cycle basically. So okay, I'm back to explain the most important figure of the study. So here comes the three part of the findings. First come the DRP1, which is the dynamic related protein 1. Then comes the SPLICs, which are actually the split GFP based contact site sensors, which will tell us about the contact between mitochondria and ER. Then comes the MitoQC, which is the mitochondrial quality control sensor, and it tells if the mitochondria is undergoing mitophagy or not. So let's first look at the DRP1. The sleep deprivation actually increases the recruitment of DRP1 to the mitochondrial surface in the DFNBs, which promotes mitochondrial fission. And this is evidenced by the relocation of the DRP1 from cytosol to mitochondria after sleep deprivation. Sleep deprivation actually leads to mitochondrial fragmentation, which is mediated by DRP1, right? And this fragmentation is a precursor to processes like mitophagy and mitochondrial endoplasmic ER reticulum contact formations. And the overexpression of DRP1 in DFNBs actually decreases sleep duration and abolishes the homeostatic response to sleep deprivation, while the knockdown of DRP1 increases sleep and enhances the recovery after the sleep. So this actually leads us to the SPLICs. The SPLICs are used to measure the number of contact sites between mitochondria and the ER, and the sleep deprivation actually increases the number of these contact sites in DFNBs, as you can see in the figure. And it facilitates the process of like lipid transfer and mitochondrial biogenesis, which brings us to the mitoQC. So the mitoQC is actually it is a ratiometric sensor that detects mitophagy. It can be also considered as clearing of the damaged mitochondria, as I've explained earlier. So sleep deprivation actually enhances mitophagy in the FNBs as indicated by increased mito-QC signals. And the significance would be that it suggests that sleep deprivation triggers the removal of all the dysfunctional parts of the mitochondria, likely as a protective mechanism against all the oxidative damage it had to go through due to the wakefulness, due to like increased wakefulness than the normal. The summary for all of this would be in relationship with the sleep deprivation would be that increased mitochondrial fragmentation, which is via DRP1, actually enhances mitochondrial ER contact size, which is measured by the SPLICs, and it stimulates the mitophagy, which is detected by the mitoQC. So mitochondria then get more elongated and less fragmented with fewer mitochondrial ER contacts and lower mitophagy activity rate as compared to the sleep deprived state. These findings actually highlight the role of how mitochondrial dynamics is very critical and the quality control in the brain's response for sleep deprivation. Okay, so we saw the effects of different proteins when they were rested and sleep deprived. But now is the part where the scientists actually experimentally induce these homeostatic responses in the FNBs. And well, they hypothesize that it should move the set points for sleep. The mitochondrial fragmentation is predicted to decrease, while the fusion is predicted to increase uh, the sleep duration and the depth and the quality of the sleep. So, how do they do it? They targeted these three factors, which is the DRP1, which we have already seen, but now it is connected to these two uh, group of proteins. The first being the integral proteins of the inner and outer mitochondrial proteins, which are termed as the optic atrophy 1. I'll call it as OPA1. And second comes the mitofusion or the mitochondrial assembly regulatory factor, which is the MARF. And this is the position, as you can see, they are located in mitochondria and would make you understand better about their role in the fusion or the fission scenario. And now comes the graphical representation of this OPA and MARF. Okay, so what is the OPA1, the optic atrophy 1's role in mitochondrial fusion, and what is the impact on sleep pressure, right? Let's look at OPA1 first. So it is a key regulator of mitochondrial inner membrane fusion, and it promotes the elongation and the interconnection of mitochondria. So, what would be the impact on sleep pressure? So, overexpressing this OPA1 in DFNBs actually increases the sleep duration and it enhances the recovery sleep after the sleep deprivation. Knockout of OPA1 actually then reduces the sleep, and the opposite, it decreases the homeostatic response to sleep deprivation. The mechanism would be by promoting mitochondrial fusion. The OPA1 increases mitochondrial efficiency and it reduces the oxidative stress, which actually then uh, lowers the sleep pressure. So, yeah, this is about OPA1. Now comes to MARF, which is the mitofusion. And what is the role? So the MARF is actually involved in the fusion of the outer membrane. OPA1 was the inner, MARF is the outer membrane, and it works in tandem with the OPA1 to maintain mitochondrial network integrity. So what is the impact on the sleep pressure? Is that overexpression of this MARF alone actually had a mild effect on sleep, but when it is combined with OPA1, it actually significantly increases sleep duration and depth, and the knockdown of MAR reduces the sleep, well, similar to the OPA1 knockdown, but at a little lesser extent. The mechanism would be that MARF supports mitochondrial fusion, which helps maintain mitochondrial health, and the same thing as OPA1 does. But the combined role of OPA1 and MARF is actually the mechanism that promotes mitochondrial fusion, and it is also associated with uh, like the counteract effect of mitochondrial fragmentation, which causes the sleep deprivation, and the balance between the mitochondrial fission, which is mediated by DRP1, and the fusion, which is mediated by OPA1, and MARF actually acts as a feedback mechanism to regulate sleep pressure. Mitochondrial fusion reduces sleep pressure by improving mitochondrial function and reducing the oxidative stress, is actually the takeaway from all these diagrams and graphs. So, to sum it all, we can say that this paper actually gave us a better understanding of sleep, and not just the traditional view of resting as a neurological reset to a fundamental requirement for mitochondrial homeostasis. It is beyond that. By identifying the mitochondrial circuit breaker mechanism, wherein the excessive electron leaked and the ROS, which is the reactive oxygen species, triggered sleep pressure, we can conclude that sleep is a critical protective response from the metabolic stress. And the key takeaways would be that sleep acts as a vital maintenance phase that prevents metabolic meltdown by allowing the glymphatic system to flush out neurotoxic byproducts like beta amyloid, and the powerhouse is actually a sensor. The mitochondria are not merely passive energy producers, they act as dynamic sensors that actually dictate the systemic behavior based on cellular health. The clinical implications of these findings that the authors and reading about it is what I think would be that it is a very massive and open avenues, like it opens new avenues for treating chronic fatigue and neurodegenerative diseases by targeting mitochondrial efficiency through a lot of possible mitochondrial and in general metabolic interventions. And I would be very excited to see what the study brings in more and like what other things this group publishes. At the end of the paper, there is actually a very interesting take. An interesting context that the authors wrote about sleep, which was the evolutionary context of sleep, the with the aerobic metabolism and multicellular life. So sleep is proposed to have evolved as an inescapable consequence of aerobic metabolism, which has a major evolutionary innovation. So what happens is that the aerobic metabolism allowed eukaryotes to maximize the free energy yield of electron transfers. It also set the stage for Cambrian explosion of multicellular life following the second oxygen revolution, which was from 570 to 750 million years ago. And they also drew the uh, parallels of power hungry nervous system and its emergence in along with it coincided with the evolution of sleep. I mean, the development of the complex nervous system, which were very highly energy demanding, they actually coincided with the evolution of sleep. So, this suggests that the sleep may have evolved to fulfill an ancient metabolic need, such as maintaining mitochondrial health and managing oxidative stress caused by the aerobic respiration. Um, the second part of the discussion was the allometric scaling of sleep. There was an empirical power law which relates the daily sleep amount to mass specific energy consumption in mammals. This relationship actually suggests that um, a fundamental metabolic uh, purpose with smaller animals is that they require more sleep due to their high metabolic rates and oxygen consumption. They also drew the parallels between the sleep and hunger. So, like in mammals, if we see the mitochondrial dynamics in orexigenic and anoxygenic neurons are actually coupled to balance energy. And similarly, in Drosophila, the mitochondrial fission and fusion in DFNBs are coupled to sleep pressure, with mitochondrial fusion promoting sleep and mitochondrial fission reducing it. I think this is gonna like this has engraved in your brain the connection between fusion, fission, and the sleep pressure is what I think. So the evolutionary statement actually emphasizes that sleep likely originated as a metabolic adaptation to the demands of aerobic respiration and the energy intensive nature of the nervous system that evolved. Over time, sleep may also have acquired additional functions such as synaptic homeostasis and memory consolidations, but in fundamental, the role is maintaining mitochondrial health and actually managing oxidative stress, which is building up during long wakefulness period of time. Thanks for bearing with me if you are still here, and I'll see you in the next paper, hopefully more interesting. Bye.